The lab side is kind of where it all becomes almost like very mysterious, because we're working with things that are invisible. We're trying to get DNA out of bacteria, and to the naked eye, we can't see any of that. So it's always kind of fun to take something like feces or soil, something that's very tangible and we can see it, and it's sometimes smelly and gross, and take it through a process that basically turns it from that to DNA floating in water.
So we do a bunch of different steps to be able to take the soil and remove everything we don't want, like the little particles of sand or clay or whatever is in there, and we pull out just the microbial DNA, bacteria and fungal in particular for this project. And then once that extraction process is complete, so I have a tube essentially of just that little piece of DNA and just like a little bit of liquid, we move over to the second part of our process, which is called PCR, and we amplify on an extreme level every DNA within that little tube. We sort of expound that by thousands and millions.
The data that we get out of our sequencing machine is really just a big jumble of A's, C's, T's, and G's, your classic DNA molecules. It's then our job to take those data and analyze them in these rather complex bioinformatic and statistical pipelines. I won't bore you with the details, but basically what that ends up telling us is a community. So if you think about the way you would analyze a community of animals or a community of plants, you have a number of species and their abundance within an ecosystem. The data that we get out is the same thing, but for microbiomes. We have all of these sequences that will match to a specific bacteria. We have their abundance and their identity. So then we have this kind of whole community that we can look at the same way you would look at the diversity of a rainforest or a prairie or anything like that, but we're just looking at this on a micro level.
And our hypothesis is that we'd have two distinct communities between a conventional and a regenerative system. Really interested in what microorganisms are shared between those two systems, but also like what are the species that are only found in those regenerative systems? Are those the species that provide those wonderful benefits that we see in regenerative systems? Improvement in soil health, improvement in plant productivity. So those species, not only the really common ones in regenerative systems, but also those really rare species. And I think it's really exciting to sort of explore the thousands of species that we get back. Like, what are those rare ones doing? These organisms, like fungi and bacteria, provide some of the most critical functions to allow our planet to function. And coming to work every day is exciting to explore this sort of unseen microbial world.